Start. Okay.
Okay. Well, thank you everyone for joining, and I apologize profusely for the rescheduling. I'm glad that I can present still this week, but sometimes things pop up that we don't always have control over. So I'm glad that everyone could make it this morning, and I'm also glad that it's being recorded. So for those who can't make it because of the rescheduling, can view it at a later time. So, a number of years ago, I presented on oxidative stress in fish in Mexico, um, in a meeting that Tanya Zenteno-Savin had organized, and that was focused on salmon. And this was in collaboration with another researcher at Carleton University named Dr. Steve Cook. And a lot of the work that I'll be presenting today is a continuation from that. I'll present the new stuff, so it won't be on salmon. It'll be on a different fish today. And the majority of this work is being done by two fabulous graduate students. One is shared between Steve and myself, who's Myra Thapar. And another student is Steve's, and it's Connor Reed. And the slides have been created by them, so I'll give that disclaimer right at the very beginning. And I'll talk a little bit more about their work. But I've given you the review article that Connor has put out on cold stress in fish, and that'll be what I'll be focusing in on today. So I'm in Carleton University. It's in Ottawa, Canada. It's based in our nation's capital. It's well less known than Toronto. So everyone from outside of Canada knows Toronto, Montreal, probably Vancouver. But we're right at the heart of the capital and Canada's government. So I'll start off today with an overview of cold shock in fish, and I'll go over the research questions that we were looking at, how the experiment was designed because it's rather complex, and two studies that we did on that and the conclusions from those studies and some of the future trajectories that we're going to take from that. So when you look at cold shock, there's a number of sources of cold shock that could occur that fish can encounter. And some of these are natural, and often these are due to extreme weather events like polar vortices. But they could also be because of upwelling of bottom water in lakes and oceans that come close to the surface. And these are standing wave oscillations. And these are going to become more and more common as our climate changes. So it's not going to be just simply global warming. You're going to have large scale patterns changes in not only weather, but also uh, the way the water moves in the oceans. And that can cause some extreme temperature changes for fish. The one that I'll be focusing on today is arthrogenic sources. So these include hydroelectric dams, hydroelectric plants, nuclear power plants, and these release cold water from bottom release dams and different reservoirs. So there's human-made sources and natural sources, and these are the types of things that fish could encounter. So fish are poikilotherms, and the general rule is that for every 10-degree temperature increase, you generally have a 2 times metabolic rate increase in fish. And so this is sort of a general rule of thumb, but this does cause stress. So temperature is the ecological master factor, so to speak, and this can influence metabolism, the immunity of the fish, various biochemical functions, growth, development, and large-scale behavioral changes as well. So I'm going to be focusing in more on the biochemical processes, and we'll talk about stress in fish due to cold. Because they're poikilotherms, their body temperature is the environmental temperature, and so they're susceptible to environmental change. there's no possible physiological thermal regulation in fish, and so the fish have to escape that through behavioral processes. And if they can't escape, then they most likely will perish in the situation. So how do fish respond to stress? There's three different layers, and the first one is a signaling layer. So this is the primary stress response. And in fish, it involves the activation of something called the hypothalamic pituitary interrenal axis. And this is the release of the stress hormones, the catecholamines, the corticosteroids. And in fish, it's primarily cortisol, which is the main stress hormone in fish. And this leads to secondary stress responses, changes in blood factors and immune function, gene and protein expression, alterations in metabolism, and the ability to osmoregulate. And this can lead up to large-scale changes in not just the whole organism, but how that organism behaves within its population and how interacting communities interact with that, particularly predator-prey communities, and how the organisms reproduce is greatly affected as well. So with the primary stress markers, you have the cortisol or corticosteroids. You can measure these primarily in blood, whole tissues, whole body, in the waste products that the fish secrete. And it's primarily cortisol, which is the main stress hormone and the main metric that's used to assess stress in fish. But there's also the secondary stress markers, and glucose is often used as a measure in blood, so alterations in glucose. Lactate buildup in blood is fairly easy to measure, changes in osmoregulation and respiratory function. The immune system, so immune changes, fish become more susceptible to disease with the stress parameters changing. And of course, the temperature stress proteins. And so this includes many of the chaperone proteins, like the heat shock proteins, which also respond to cold. And they're the main factors that most look at when they look at cold shock, even though they're termed heat shock proteins. And this can be measured in blood and tissues as well. For this group, this is where oxidative stress comes in. And so reactive oxygen species can be measured, antioxidant enzyme function, changes in lipid metabolism and lipid profiling can be done, alterations in cell membrane, especially with cold, often occur. And the whole field of lipidomics looks at the changes in the phospholipids in membranes with cold temperature changes. Tissue death, cell death, mainly measured by lactate dehydrogenase release, but there are other methods of looking at tissue damage in tissues. And then there's all the omics approaches that can be taken. And with fish, it's primarily genomics that have been looked at. I would like to move the field into proteomics because I'm a protein person. But there's very little work that's been done in that area, and I'd like to sort of move in that. Oxidative stress can affect not only the primary stress response, but also the secondary stress response. And changes in the brain will alter the release of the corticosteroids and catecholamines. So reactive oxygen species damage can cause neurological changes, and this affects the hormonal release that you often see in the primary stress response. And you have the elevation of genes and proteins mitigating oxidative stress. You also have changes in membrane composition to maintain membrane fluidity under colder conditions. So these are a lot of the changes that you generally see in the generalized stress response. And cold stress has been shown to increase reactive oxygen species and oxidative stress. You do have increased lipid peroxidation as a measure of oxidative damage. We have new probes now to measure lipid peroxidation, including ones that you can use directly in cells that are BODIPI based. And so there's a lot of tools out there now new tools to look at lipid peroxidation as an indicator of oxidative stress. And the presence of a number of different antioxidant enzymes do change in fish, even with cold shock. So you'll see a lot more changes in heat shock, for instance, than you will in cold shock, because it's a more extreme case. And in general, with cold shock, you're trying to shut down metabolism. So transcription and translation generally are repressed. And so you see fewer changes with cold shock than you will with heat shock. And so the gene changes that can occur generally are with the chaperone proteins. There has to be more work that needs to be done on gene work. There's a number of different studies and a number of different fish that look at gene expression patterns and how they change with the stress response. And this is just an example of one that has been looked at in the turbot, where you have an example of the heat shock proteins actually increasing with colder temperatures. And you can see some of the major changes in heat shock protein 70 occurring in the brain and the head kidney in the fish, but also in the intestine, in the straight kidney, and some changes in the gill as well. So all of these go up when the temperature goes down. And you can see that this study tested all the way down to 1 degree Celsius. That's pretty extreme cold for a fish, and for a period of 9 hours. So back in 2008, there was one review article put out by Donaldson and Steve's group on cold shock, and they defined what cold shock meant to fish as an acute decrease in ambient temperature that causes the potential for rapid reduction and a rapid cascade of physiological and behavioral responses. And so they did a large review on some of these physiological and behavioral changes in fish. But since so many more studies have been done, the review had to be updated. And this is where Connor's work comes in. So a lot of these are Connor's slides, which he presented to a group on this large expansion of the cold shock review in fish. And so this is the updated review that I've provided to everyone. And it highlights various potential biomarkers, as well as gives some very good practical aspects and applied perspectives that cold shock will mean. So it's a rapid decrease in temperature, and this is taking a Canadian example where we look at one of the Great Lakes, Lake Ontario, that's present in Canada, and it shows you Can everyone still hear me?
Yes.
Okay, I just wanted to make sure that we didn't lose anyone. Okay, so the top graph shows the temperature changes going into the winter months, and you can see it's quite a precipitous drop from 18 degrees Celsius down to 4. But if you take a look at the bottom graph, this is in the August months, and this is still considered to be summer for us. And you can see a precipitous drop from 24 degrees Celsius to 8 degrees Celsius in the surface temperature of Lake Ontario. And this happens during one period in August. So this could be due to upwellings from the bottom of the lake, or it could be due to anthropogenic purposes. A large release of cold water into the lake will cause this as well. And this is a pretty extreme temperature change for fish. So this is one example of where you can get either a natural or a man-made occurrence to temperature changes in lakes. So what our study focused on was actually nuclear power plants. And nuclear power plants take in water from the Great Lakes, and they have a screen that covers the water intakes into this. And fish become impinged upon that. And the death that's seen in these fish that become impinged on these grates is due to cold shock, actually. So the question becomes, is the death of the fish due to the impingement or is it due to the cold shock itself? And so we started to work with partners with different groups. One of them is Kinetrix, and the other is the Nuclear Power Plant Owners Group called CANDO, and to look at cold shock in fish. And so it's a little bit of teasing apart all of the stresses that could occur in fish with this type of impingement. So did the fish die due to impingement, or was it due to cold shock or illness or possible exposure to air? A lot of these stresses co-occur together, and the effects of each individual shock or stress is a little bit hard to tease apart in some cases. You don't know whether the responses that you see are due to one stress or another. So the reason why we undertook this study is to advance the field of thermal stress. We wanted to isolate some new stressors molecularly and detect some new biomarkers, create some new tools for use in the field for fish studies and stress response, as well as to inform policymakers and those that manage nuclear power plants and environmentalists themselves. So there's a larger political aspect to this work as well. So this is a large call out to our grad student. This is Myra, and grad students are critical and key to all of this work. And Myra's an exceptional individual who can run SDS page gels in the lab one day, and then the second day jump on a boat and be catching fish out in the Great Lakes on another day. And so this is Myra in both cases. So it's rare to find these individuals who are good at both field work and the lab work, and Myra's one of the great grad students that's an exception to the rule. And so this is the perch that we work with. It's called the yellow perch. It has a number of different names, but its scientific name is Perca flavescens. And this is our main study fish for this particular research. So our research questions were, what does the cellular response to cold shock stress look like in fish? How does it differ from other stressors? And the other stressor that we were looking at was primarily hypoxia. So that's basically air exposure in fish. And how does the intensity and duration of the cold shock affect how fish respond to the stress itself? And so we came up with a number of hypotheses. And we hypothesized that cellular stress will be affected by cold shock, that it will differ from primarily hypoxia, and that the intensity and duration of the cold stress doesn't influence and that it does influence the molecular responses by the fish. This is Myra's experimental design. So what you have here are black boxes, which are acclimation boxes. The fish were placed in these boxes where water was circulated through. So you can see the chilled reservoir and the circulator on the right, and the individual boxes that the fish were placed into on the left. And most of this was created from parts that were purchased from the local hardware stores. So that's what you really need. You need creative and inventive grad students to be able to put all of this stuff together on the fly. And so this is the internal workings. So the fish were placed in the sensory deprivation chamber, basically a black box, and it had a recirculation pump. a flow delivery pump, and 2 aquarium heaters, an air stone to keep things aerated, and a temperature monitor as well. And so we treated fish with various treatments, which I'll talk about in a bit, but we isolated RNA from fish tissues, primarily liver and muscle and gill. And some of these were profiled on this stress transcriptional profiling chip, which was developed by the University of Windsor in Ontario, Canada. And this is part of the GenFish project, which is a project that is part of Genome Canada that we obtained funding from. So this fish has about 28 different genes on it, all related to stress. These are the genes that were tested. So there's stress response genes, there's endocrine disruption genes, growth and metabolism, immune function, and a lot of the ribosomal proteins were used as endogenous controls in this case. So this is what the profiling chip looks like when you actually run the chip itself. It's a large series of genes that does real-time PCR all at once and profiles all of these stress-related genes simultaneously. So it's a large amount of data, and you have to go through a lot of data. And the analysis can be quite tricky sometimes. So for our first study, we asked the question, does cold shock differ from other stressors? So does it differ from hypoxia? Does heat shock differ from hypoxia? So often these two stresses are encountered together. Fish could be caught in nets and say remain in the net, which is a definite temperature change, but you also have hypoxia. And some fish are considered to be bycatch. So these are species that are not commercially, wanted. So these are tossed back, and the question becomes, will that fish survive after the bycatch has been released, or will it perish because of the stress that it has undergone due to the hypoxia and the extremes of temperature that it's being exposed to while in the net? And so this was the experimental design. We did look at liver, gill, and muscle. We collected fish directly from the lake, and these are our field samples. These were euthanized immediately. And then we put fish in the experimental setup. Myra was doing all of these experiments at the Queen's Biological Station that is close to Carleton University. So controls were euthanized immediately, and they were placed at 24 degrees Celsius, which is the average lake temperature that they were in. But they were also exposed to air. This is the hypoxia heat shock, which is 32 degrees Celsius, and 3 temperatures of cold shock. So 8 degrees Celsius, 12 degrees Celsius, and 16 degrees Celsius. And then they were euthanized. And so RNA was extracted, and we looked at the gene chip. We did a number of other gene studies as well, but I'm only going to present the gene chip data today. And we analyzed the data using the LiveAct method, and we also did a principal component analysis, which I'll have to describe what that means when I show the data. And so for all of these different stress response genes, there were 3 of them that exhibited poor amplification, so they weren't able to be detected in tissues, so we left those out. But there was only one gene, which was matrix metalloproteinase, which didn't have any changes, but all other genes did have changes and responded to the stress response. And these include various things like hydroxysteroid dehydrogenase, which you can see goes up in air as well as in all the cold treatments. But also all of the others were statistically significantly different as well, with the exception of MMP2. And we did a principal component analysis, and this takes a bit of explanation. So principal component looks at the variability that you can have in the data. And principal component one is the component that has the highest variance. PC1, as we call it, has the highest amount of variance percentage amongst individuals. And then PC2, or principal component 2, has the second highest variance, and so on. So this graph on the right shows the cumulative percentage of a variance that you could have in the components that you're looking at. And then the principal component numbers are shown with the black line. And these decrease as you do iterations of the analysis. So we really focused on PC1 and PC2. And this causes the genes to cluster when you graph one versus the other. And so this is a PC, principal component analysis graph. And the thing to note is that when you have genes that cluster together, these are positively correlated genes. When they're 180 degrees separate from one another, they are negatively correlated. And when they're at 90 degrees to one another, there is no relationship between the genes. So one of the things that to note with the stress response genes, if you look on the graph on the far right, is that the heat shock, which is shown as the green dots, seem to cluster together quite nicely. So heat shock is a little bit easier to work with than cold shock. It has a lot more obvious changes that occur, and these tend to cluster in the PC scores. The other difference that seems to be clustering is those that were captured directly from the field. And so these seem to separate out from all the others because the other controls were acclimated in these sensory deprivation boxes along with all the other treatments. So that makes sense. So these are the endocrine disruption genes, and these include genes like estrogen receptor and androgen receptors. So, parametric and non-parametric tests found statistical significant differences between treatment groups in ESR2B and ESR1. These are the estrogen receptors. So they found differences between field and the 32 degrees Celsius treatments in ESR2B. And then they found differences between field and the 16 degrees Celsius treatments in ESR1. So this is the way that the principal component analysis panned out. And you can see that there are fewer relationships when you're looking at endocrine disruption within fish because the dots are kind of intermixed amongst one another. The angles are mostly at 90 degrees. So there was little relationship between those endocrine disrupting genes. When you look at growth and metabolism, there were differences found in treatment groups for IGF. These are the insulin-like growth factors, and the cathepsins. So post-hoc treatments found most of the difference between field and IGF-1 and CTSD. And then differences were also found between the heat shock and all other treatments with IGF-2. So this shows the changes in heat shock. But again, the principal component analysis showed an intermix of dots, and then they weren't positively correlated with one another when you looked at the treatments. So there's little relationship between them. They were all at 90 degrees south, 90 degrees to one another. The immune function genes found also differences. So post-hoc differences were found between 32 degrees Celsius and all other treatments for TAP BP. And this is a protein called papain. The major histocompatibility complexes showed differences between field and all the others. And then the 8 degrees Celsius treatment was found in calcium calmodulin proteins. And this is one that I'd like to focus in on later on because CAM actually showed the most consistent changes, especially with cold shock amongst the fish. And so when you look at the principal component analysis, everything clusters together on one side. And of course, heat shock, again, seems to be well separated from the others. But there were interesting findings in the immune function genes, which I'll talk about in just a minute. So the summary of study 1 was that most differences were occurring between field and all the other groups. And this makes sense in terms of how the fish were acclimated to the experiment. So the true controls were the ones in the sensory deprivation boxes, but not treated with anything. The coldest treatment had the least amount of relative fold change, and this makes sense because with cold, the organism is shutting down transcription and translation, and it spends less energy on protein synthesis in general. But despite that, there were 5 genes that seemed to be potential markers for cold shock. And one of these was calmodulin, which I'll talk about in a minute. But also the heat shock proteins were also seen to be positively correlated with one another. And the androgen receptors and the estrogen receptors also seem to be positively correlated with one another. So those were the main findings of this first study. And the second study looked on time, had a time influence. So we wanted to understand what time had, or what influence time had on cold stress and the molecular response. So the design of this experiment was to take fish at 8 degrees, 12 degrees, and 16 degrees Celsius in our experimental setup and euthanize about 4 per hour for a period of up to 5 hours. And this is not 4 simultaneously. This could be 4 within the hour. So the design, or the graphs, look somewhat like this, where the number of fish that were sampled are shown in the blue dots, and each dot represents a single fish. And this is the time frame. So basically, you have a whole profile over the entire time course of 5 hours between 8 degrees Celsius, 12 degrees Celsius, and 16 degrees Celsius. And again, we did our chip analysis, and we did the LIVAC method to look at fold change, but we also looked at linear regression to see what those changes meant. And so you can see that some of the changes here that occur, what's interesting is that heat shock protein 7D seemed to have a positive correlation at 16 degrees Celsius, but really seemed to not have much of a change at all at either 12 or 8 degrees Celsius. Some genes were consistently high within cold and came up to very high gene expression during the cold, and this includes SERPIN-H1. And but quite a few of them had negative correlations as well when you look at the data between the different temperatures. For the endocrine disruption genes, the estrogen receptors were often very high. We saw positive relationships with one of the estrogen receptors at 12 degrees Celsius. But again, those differences were less when you looked at it at 8 degrees Celsius in general. Growth and metabolism genes, again, mostly negative correlations. There were a few changes in the insulin growth factors, but you can see some negative relationships here in some of these. And then immune induction genes did have changes. One of the more consistent changes, although it's not seen quite as well here, was actually in CAM. calmodulin. And you can see that some genes like TAP-BP are very high in consistently with cold shock, especially at 12 and 8 degrees Celsius. And but relatively overall, the time course, they had relatively little change. So the summary of this study was that each gene responded and with time difference, and certain genes were more elevated at different cold temperatures compared to others, despite the fact that they may not have had change over the entire time course. They were consistently high. And SERPINH1 gene was elevated at both 8 degrees and 12 degrees Celsius, while CAM seemed to be expressed at higher amounts at the 8 degrees Celsius. There was also hydroxysteroid dehydrogenase increased by a large fold change in the 12 degrees Celsius, but primarily in the 12 degrees, and not the other two temperature time courses. And heat shock proteins and estrogen receptors were also found to be upregulated, but this fold change was primarily seen at 16 degrees Celsius. So, concluding, the specific changes in the message RNA were seen to be temperature and time specific. Again, a lot more changes are seen in heat shock and often hypoxia than what you do see in cold shock. There's a lot of variability amongst fish, and so each treatment we had to do the principal component analysis to tease out changes within that variability. And calmodulin was identified as the most promising biomarker for cold shock because it seemed to be consistently changing with the treatments, not only the temperature regimes, but also over time. And it seemed that 8 degrees Celsius was the best inducer for calmodulin, so the more extreme cold shock temperatures. And relationships between the specific genes were isolated, but these studies are still ongoing, and we're still working out some of the differences between those. So calmodulin is an interesting protein in that it's involved in calcium signaling. It's actually a calcium sensor protein, and this corresponds as an intermediate protein between calcium binding and other proteins and other functions that are calcium dependent. So it's kind of the bridge that is the gap between calcium sensing and calcium affected functions. So this is very interesting, and we were sort of scratching our heads at this one for a little while, but we actually came across two papers where they take fish calmodulin and they introduce it into plants, and this improves their cold tolerance. So it may not be quite as far-fetched and there may be something that calmodulin is doing, and calcium in general, during cold shock. And these actually took calmodulin from an Antarctic ice fish to use in plants. And they found that cold tolerance was increased. It improved chilling tolerance in various tobacco-like plants as well. So these are two different interesting studies. Has interesting implications, but we're still working out what the role of calmodulin is doing in fish during cold shock. So the future trajectories are to actually test temperatures closer to the lower thermal limit. We find larger changes closer to that lower thermal limit. So it's a fine balance between, not stressing the fish out too much and, approaching those extremes so that we can see larger changes in gene expression. And we have to consider protein expression and how it relates to gene expression. And so I would like to take this in the proteomics direction. There's two challenges to that. One of them is that the antibodies to fish proteins are mostly created in mammals and don't cross-react very well. So we always check the binding sequence for antibodies to see how closely the mammalian ones are related to fish. And the other thing is that when you're trying to identify them with, say, mass spectrometry, the databases in mass spectrometry are built on mammalian species and may not always identify proteins from species outside of that. So those are two of the challenges that we have to overcome when we look at protein expression, but we're taking this to the 2D gels and to isolate individual proteins, which may change with cold shock. And teasing apart multiple stressors, because as I mentioned before, these often occur not alone, but in tandem with one another, is a big challenge. So we're trying to figure out how multiple stressors encountered at the same time can be teased apart in terms of the stress response. And we're also looking for new biomarkers specifically for cold stress in order to be able to implement these to diagnose whether a fish has undergone cold stress in the field. And so we're trying to develop field usable tools so to determine if the fish has undergone cold shock. And so I cannot do this alone. We have a number of different collaborators. Steve Crook runs a huge group here at Carleton University of grad students and all focused on fish. And so a big thank you to the Cook Lab members who have helped Myra and have helped Connor in all of their projects. You'll notice that the Cook Lab members also include members that have the last name Cook. And I think these are Steve's children who he pays to go out into the field and catch fish. But it also is the Can Do Owners Group and the Bruce Power Plant groups, and also the GenFish project collaborator, Dr. Kenneth Jeffries, who is in Manitoba, and Myra has done a lot of gene analysis in Ken's lab in Manitoba, as well as working with the gene chip at the University of Windsor. So a big thank you to all of our funders, a very big thank you to the two graduate students starring in this, Myra and Connor. And I can take any questions if you have any.
Oh, thank you, Bill. It's very, very nice study. It's a lot of results. I would like to start with a question.
Mm-hmm.
Since you start your presentation with stress hormones, so did you guys do correlation between your results in all these conditions, or at least some of these conditions with stress hormones?
Yeah, that is something that we didn't look at specifically. Cortisol analysis is done at the other university that's in town here. It's an HPLC method that they use. We didn't look specifically at cortisol in these fish, but that's something that can be done, and to see what the cortisol levels are in fish. In some of our salmon work that we've done with Steve, cortisol is always measured. And with those, the salmon that spawn in British Columbia, there's stress all the way up to the spawning site because they migrate from the ocean to freshwater to spawning sites. Shock. Generally, what has been found in the past is that the corticosteroids do not change quite as much, which is interesting. There seems to be a slowing down of metabolism, a slowing down of the stress hormone release. And in other studies, they've found that cortisol is maybe not the best indicator for cold shock.
Another question. Did you measure or plan to measure expression of some antioxidant enzymes?
Yeah, so that is something that I would like to do. You notice that with the gene chip, there weren't many antioxidant enzymes on it. If I were to design the gene chip, I would include a lot of those, but I didn't design it. So, but I would like to look at a lot of those and see what their expression does. Often cases, the genes sometimes don't change, but then you generally see large changes in activities. So what I'd like to do is I'd like to do the assays for the enzyme activities, because it's a little bit more like reading the last chapter of the book, right? To see how more functional they are. There could be changes that are post-transcriptional, changes in proteins. We tried a number of different Western blots with some of these, but often the human-made antibodies are not a good cross-reactor with fish. So we need more fish antibodies.
Yeah. Yeah, the old assays, they're still working.
Yeah. Yeah. So it's good.
Yeah. Okay, everyone, please ask questions.
I have one question.
Mm-hmm. Hi.
Hi, Bill.
Hi, how are you?
Hello?
Hello. We're starting to...
I'm okay. A long time. It's a long time I don't see you.
Yeah, I
Can you
need
listen to me?
Yes, I can hear you. I can hear you. Can you hear me? Sometimes the connection
Yes,
is not the
yes.
greatest.
It was nice to see your presentation. Yeah, it came out of poor network conditions.
Yeah.
Bill, my question is, in the study one, how long the fish stay in the various temperatures?
Yes, that's a good question. I believe that it was 24 hours. It was a bit longer in terms of study one.
Okay.
So this was a longer time course.
Okay.
If Myra is on, I see Myra on, she might be able to better answer this question.
Yeah, so is everyone able to hear me?
Yes.
Okay, perfect. So it was 24 hours at the 24 degrees Celsius temperature, which is the control temperature. And then for any of the experimental temperatures, it was 1 hour for the first experiment.
Okay. And I look at the genes and that were increasing in study one. And one of the genes is an enzyme that catalyzes interconversion of inactive cortisone to active cortisol. Could this be an indicator that cortisol is increasing? Is the gene called HSD11B1?
Yeah.
Yeah, so fish, I believe that if I remember correctly, fish don't have that. Wait, there is, I believe, so HSD11B1. It was like the converting enzyme that you're speaking of. It's like that protein. It's not necessarily the protein. So the gene codes for like an HSD11B1 like gene. It's not the same. But yes, I did go down the same avenue that you're suggesting, and I did do some research on that. So If it is, then it's really cool.
Yes, yes, for
Yeah.
sure. Okay,
Being
thank you very much.
Thank you. It's interesting that, yeah, it was highly activated, and it would suggest that cortisol is playing a major role. So cortisol should be looked at in this particular study. So I don't know, did we get blood samples for that, Myra?
No, we did not.
Okay, we were taking tissues mainly.
Yeah, it was on a time crunch. So with every experiment, what we tried to do was to be able to dissect all the samples at the 1 hour mark. And with the boxes, we're limited to 9 fish per box. And that gave me a total span of 10 minutes to dissect and get all these organs. I would like to add on a little blood element eventually and like reiterate the experiment and get blood from these fish. And if I do a blood work study, then I could also release these fish, so it wouldn't have to be a mortality either. But yeah, it was just not feasible to get blood and dissect within the time frame when I was working on it.
Working in the field has certain challenges. So Myra's become very adept at dissection of fish very rapidly in the field, maybe on a boat sometimes.
Yeah, getting the weight of a fish on a boat is really tough.
Yeah.
Yeah.
Any other questions?
I have a question to Myra, but first I would like to congratulate you for your work and to invite you to participate in our club, to attend the next meetings, the next seminars. And I have a question. I'm looking at this slide, number 30. In field conditions, what is the temperature in field conditions? So it depends on the day?
It depends on the day, but it depends on the time of year. So all of these fish were collected at different times because there were so many of them. I believe the first year I went in, there was 400 fish that were collected, but there's like a turnover event that occurs, and water temperatures are usually, so all these fish were collected between June and July. And there's a water turnover event, and during those 2 months, the water temperatures are around 24 degrees Celsius. However, perch, they like to live at about 14 feet deep. Around that time, like the summer I was collecting it, I mean, the study isn't perfect, but there's a lot of storms going around, and I do believe the water temperature was lower, but just to keep it consistent with the rest of the study, we acclimated these fish to 24 degrees Celsius. But if I were to redo this, I would probably be collecting water temperatures myself. It was just difficult to do that because I wasn't doing all the fishing. And like Bill mentioned, some of Steve's kids were doing it, and these kids are so good at fishing, but I don't think they had the time to be collecting temperature. But yeah, these are just like the 24 degrees Celsius really came based off of research and estimates of what the water temperature should be like around that time of the year. So yeah.
Our control was 24.
Yeah.
Yeah, thank you. I had a very little experience of catching fish in the wild in the Amazon, and I started a project to see what happened to fish in different areas of the Amazon, areas where oxygen is kind of normal for the time of the day, for the season, and other areas that go hypoxic. But the thing is, the fish, they're always swimming. So you never know how long the fish is in the area where it's hypoxic or normoxic. And there are several variations, not only I mean, when you look at different areas, but in depth. Yeah. And so it depends on the time of the day. So it's just kind of impossible to to do this. So that's why we're working with animals. They don't move, so they're like oysters.
Yeah,
So we can.
Marcelo has hit upon the biggest challenge of doing field work, and this is what Steve's group has found. You know, you have different individual fish exposed to different individual temperatures, and so the variability can be quite large. You know, when you're trying to do field work and you're trying to do the assessment from fish caught in the wild, it is very challenging. So.
Yeah, it becomes really challenging because, you know, from like the scientist's mind, I would want to, raise these fish, know their history, know what background they came from, and then introduce them to all these conditions and see what changes. And I'll probably get more of a clear understanding of it. However, it then becomes challenging to be able to relate it to wild caught fish. Fish in the wild versus lab fish react so differently to anything. But then again, we encounter the challenge of using field fish, but not knowing their history. However, there's high individual variability where each individual is doing its own thing. And, yeah, it's just, it's challenging either way, I'd say.
Yeah, it's very difficult. So that's why we gave up. Because the place for catching is far away in the Amazon, and you see you don't have many chances to go there and repeat the catching. So we had to stop the project, but it's nice to know that there's someone else trying.
Yes. Any other questions? I may get some questions later on. So if this is being recorded, I highly encourage people to email me any questions that they have if they're watching the video later on and have questions. My email is right at the bottom there. It's just bill.wilmore@carleton.ca. So feel free to send me a quick email or Myra. It's the same email for Myra, so it's myra.thapper@carleton.ca, and so we can answer any other questions that pop up after this.
Okay, so thank you, Bill.
Thank you, Marcelo.
Myra. Yeah.
Thank you for inviting me to give this talk. And again, my apologies for not giving it Wednesday, but I'm hoping that everyone can at least see the video of it.
Yeah. The idea is to show people the results and stimulate others to follow these steps, and so thank you. It's nice to see you. So we keep.
It's great seeing you again.
Yeah, you haven't changed since the last time, 2019. So this time, the Ken Star didn't show up today.
Yeah. So I'll have to relay to him the video link.
Yeah. Okay, thank you everybody for attending the seminar. Again, thank you, Myra, Bill. So see you guys in the next seminar. So we hope we'll still have one seminar left for this year.
Okay.
So I'm talking with some people, see which person will be next. Okay.
Well, thank you for the invite. Thank you for Marina for setting this up again, and thank you everyone for attending.
Yeah. Bye-bye.
Bye-bye, everyone.